Assalamualaikum and hi, my name is Nurisha Fazlin binti Ilyas Faza and I'm going to present my findings study on antioxidant activity and total phenolic content of stem buds and leaves of Murraya koenigii. So welcome to my presentation where this is the item that I'm going to explain in this video. The one that we are familiar with is modern medicine where it was used widely in the healthcare industry. Examples are drugs and surgery. Traditional medicine was very popular back in the days, however, nowadays it gained popularity back. Many studies stated that modern medicine is more effective. So why has traditional medicine is gaining attention back? Because, however effective it is, modern medicine has negative side effects. Of course, all medications could have side effects, but to compare both of them, the traditional one was said to be safer together with the increasing demand, affordability, and easily obtainable, making the traditional medicine popular again. So the famous traditional medicine is the herbal plants. This is why this study will provide useful data on these herbal plants named Murraya koenigii, also known as the curry leaf tree, and yes, we are very familiar with this plant. So this research study on antioxidant activity and total phenolic contents of stem buds and leaves of Murraya koenigii. The objective is to compare between those both parts in terms of their antioxidant activity using DPPH radical scavenging assay and also their TPC by using the falling Q culture method. In the literature review part later on, I will explain these principles of these two methods. So more research on the leaves has already been done and published, but there's been little research on the stem bark, which is why the study was conducted on both parts so that they can also be compared. It was said that both parts were rich in antioxidants, so the data that I'm going to present later on will help improving this statement. Another reason, like I said before, is due to safety concerns because long-term usage of modern medicine could have negative side effects. This study is important because Murraya koenigii was said to have a high concentrations of antioxidants. Second, these findings could have been drug development to make antioxidant-based drug formulations, as it was said to have the potential to prevent and treat serious conditions such as cancer, stroke, diabetes, Alzheimer's, and many more. In simple words, treating diseases caused by free radicals. Murraya koenigii actually came from India, but due to immigration history, now it can be found in most Asian countries, including Malaysia. It was commonly used in food preparations and medications. As stated by Handra and his friend in their study, this plant could treat humans internally and externally. For example, helps with indigestion, kidney problems, insect bites, wounds, and many more. The phytochemicals in the plant are the things that contribute to curing all of the conditions mentioned. This is why the study extracted and screened phytochemicals where previous studies mentioned that phenolic, terpenoid, flavonoid, and alkaloid are some of the common ones present in Murraya koenigii. We need to understand that what are antioxidants first. It is compounds that have the ability to stabilize or neutralize free radicals without becoming unstable itself. How? By donating electrons. So what are free radicals? It is a molecular compound with unpaired electrons that are capable to exist independently. Is it a good thing? Well, actually, high levels of radicals could lead to oxidative stress, which could damage cells, fatty tissue, DNA, proteins, and cause several diseases. So in simple words, the imbalance between free radicals and antioxidants in the body is what leads to oxidative stress. In antioxidant activity, we use DPPH radical scavenging assay. DPPH is a stable free radical that does not dimerize like most other free radicals. It is deep purple color due to the electron delocalization. So when mixed with extract, the antioxidant in the extract will scavenge the DPPH free radical, leading to the mixture solution color changes from purple to yellow with a decrease in the absorbent at 517 nanometer. Following Kikati method uses FC reagent, which this reagent capable to reduce the phenolic groups, resulting in the production of blue color in the mixed solutions, and it can be measured spectrophotometrically at 760 nanometer. This flowchart is a summary of the methodology in this study. The stem bark and leaves of Murraya koenigii were collected, washed, dried, and ground into powder form. The first step was consecutive soaking using 3 solvents, which were hexane, ethyl acetate, and methanol. The extract obtained was evaporated using a rotary evaporator to obtain liquid extracts, and by the end of it, there were 6 types of extracts, which are hexane, ethyl acetate, and methanol extracts for both stem barks and leaves. So first we do thin layer chromatography, TLC, to separate the compound presence and choose the best ratio for each extract. The development system was prepared by mixing 2 solvents, hexane DCM for hexane extract, EA DCM for ethyl acetate extract, and methanol DCM for methanol acetate. DCM stands for dichloromethane. So the plate was observed under a UV lamp to know which ratio gives the best preparations. Then it undergoes chemical visualization using those chemical reagents stated. So since the DPPH reagent shows the antioxidative of the extracts, hence, only the HSC plate that gives a positive result with it will proceed with the next analysis, which was dot blood assay. In that blood assay, also use the assay where serially diluted samples from 100 to 0.024 mg per ml were spotted. Then it was sprayed with 0.05% DPPH, and a yellow spot on the purple background was observed. Moving on to DPPH radical scavenging assay, each crude extract solution ranging in concentration from 400 to 12.5 microgram per ml was prepared first. then followed by the preparation 0.004% DPPA solution. Then 1 ml of extract from each concentration was mixed with 3 ml of DPPA solution, shaken vigorously and placed in the dark for 30 minutes. Then the absorbance at 5.7 nanometer on the UV visible spectrophotometer was recorded. And lastly was the following decanting method, where 1000 ppm concentration of each extracted sample was prepared and mixed with 2.5 ml FC reagent and 2 ml sodium carbonate solution, and the standard gallic acid solution with different concentration from 1000 to 31.25 ppm was prepared and the same procedure was applied. And all the mixture solution was incubated for 30 minutes. And the blend solution was also prepared by mixing only the FC reagent and sodium carbonate solutions. Then the absorbance at 765 nanometer for gallic acid and extract sample was measured compared to the blend solution. So now finally we're going to take a look into the important part of the study. For phytochemical screening, we use TLC method. The analysis is qualitative because it only reveals what compounds were present. The results were already summarized in table 4.1. So only leaves hexane possesses all three types of phytochemicals, which were phenolics, terpenoids, and alkaloids. Phenolic was the only compound present in all sample extracts. The ones that marked with stars were the only extracts that were known to be anti-oxidative in this analysis. Next, I'm going to discuss the antioxidant activity of Murraya koenigii, and let's see the semi-quantitative result obtained from the dark blood assay first. Those four were the one that was proven antioxidative in the phytochemical screening before. It was compared with the standard ascorbic acid. The one that gives the lowest concentration is the best DPPH free radical scavenger among the extracts because it indicates that they could exhibit antioxidant activity even at low concentrations. From table 4.7, we could see the hexane and methanol extract of leaves yielded the lowest concentration, followed by leaves, ethyl acetate, and stumbark methanol. The leaves hexane extract observation correlates with the antioxidant screening because it shows all positive results indicate that it contains many phytochemical compounds responsible for its antioxidant activity. While for leaves methanol extract, it might be due to high amount of phenolic content present. Leaves ethyl acetate extract contains only phenolic and terpenoid compounds, but it was assumed that the terpenoid compound contributes to its activity because the acid screening for phenolics only show little dark blue spots, meaning the phenol content might be low. So moving on to the next quantitative analysis using DPPH radical scavenging assay. The graph of the percentage of scavenging against concentration was plotted to calculate the IC50 values. The lower the IC50 value, the better it is for the extract to scavenge DPPH radical, indicate that it has stronger antioxidant activity. So the best scavenger for stumbark was ethyl acetate extracts and for leaves was methanol extracts. Let's dive into this data deeper, whether they correlate with the TLC screening and dark blood assay or not. So stumbark ethyl acetate extract did not demonstrate a positive result on TLC. So supposedly it is non-antioxidative, yet it is the most antioxidative among all stumbark extracts given the lowest IC50 value. The extract only contains phenolic and the TLC screen does show very obvious yellow spots. So for now we could assume that the phenolic contributes to its activity, while for leaves methanol, the result was consistent with the TLC analysis. That it proved it was antioxidative and supported by the dark blood assay results given the lowest concentrations of DPPH inhibition. So let's compare in terms of its radical scavenging activity with different concentrations. Figure 4.4 shows that the radical scavenging activity of all stumbark extracts rises in a concentration-dependent manner, meaning the higher the concentration of each extract, the greater the radical scavenging activity, except for the ethyl acetate extract, which reduced slightly for 400 concentration. However, the value is not particularly significant, which could be due to systematic instrumental error. Ethyl acetate extract dominate the percentage radical scavenging activity, followed by methanol and hexane extract, but methanol extract surpassed the ethyl acetate extract at 400 concentrations. Figure 4.5 shows that the radical scavenging activity for leaves extract does not follow the concentration-dependent pattern. All of the extract's activity started to increase in concentration but then began to decline at around 100 to 200 microgram per ml. The decreasing activity for hexane and methanol was not that significant, but ethyl acetate decreased drastically at 400 concentration. So for concentration ranging from 12.5 to 50, ethyl acetate extract outperform methanol and hexane extracts. However, the methanol extracts start to take place at the concentrations of 100. The TPC value was calculated using the gallic acid standard curve. As we could see, methanol extract had the highest TPC value for both stumbarks and leaves. Even though stumbark methanol does not have the best antioxidant activity among all extracts, but the higher TPC value indicates that phenolic attribute to its activity. The high TPC value for leaves methanol extracts explain why it was the best radical scavenger of all the extracts, as proven by dark blood and radical scavenging assay. Hexane extract for both stumbark and leaves was not detected, indicating that the phenolic content was too low. And based on theory, since phenol is a polar compound and hexane is a non-polar solvent, there should be no phenol in the extract, which correlates to the TPC data in this study. We are already at the end part of the presentation, so let me conclude the findings of the study. The assay showed that hexane and methanol extracts for leaves has the lowest concentration for DPPH inhibition at 0.048 mg per ml. In DPPH radical chemistry assay showed that the stem bark ethyl acetate extract and the leaves methanol extract had promising antioxidant activity with the lowest IC50 values as stated. And the highest TPC values for methanol extract were found in stem bark and leaves. As you can see, leaves of Murraya koenigii from methanol extract are a promising source of antioxidant agents, as the data is consistent between all analyses conducted in this study. Given this high antioxidant activity due to its phenolic content as proven by its high TPC value. Further research should conduct phytochemical screening tests to detect as many as possible components responsible for antioxidant activity and also conduct FTIR spectroscopy to determine the functional groups of the compounds in the foods. Thank you for your time listening to my presentations and have a good day.